Hello, my name is Tatiana Joaquin, and today I will continue the investigation on the enzyme lactase. In this video, we will explore the catalytic mechanism, catalytic strategy, and an effectiveness experiment of the enzyme lactase. Lactase is a beta-galactosidase enzyme, which means it catalyzes the hydrolysis of a substrate with a beta-glycosidic linkage. Lactose is the substrate, and it contains a beta-glycosidic linkage. The quaternary structure of lactase shown here is made up of 4 identical subunits. Each subunit is a chain made of 1,023 amino acid residues. The structure shown is in complex with lactose. This enzyme is important in the digestion of lactose. Without it, lactose cannot be digested fast enough and lactose intolerance occurs. A common assay to determine the enzymatic efficiency uses an ONPG analog instead of lactose as the substrate. When ONPG is hydrolyzed, it produces ONP, which gives off a yellow color that can be measured with spectrophotometric analysis. Data from the assay is used to create charts and graphs, which then help us to determine kinetic parameters such as Km and Kcat. Here is an image depicting the general reaction which lactase catalyzes. This is a glycosidic hydrolysis reaction, which is a reaction that breaks a glycosidic bond. Studies suggest that the enzyme lactase helps to initiate and speed up the hydrolysis reaction. There are 2 proposed mechanisms when it comes to the catalytic mechanism of lactase, and they somewhat overlap. The first mechanism suggests that lactase initiates hydrolysis when the glutamate nucleophile, glutamate 537, on the enzyme attacks lactose from the axial side of the galactosyl carbon in the beta-glycosidic bond. Details on how glucose separates still have yet to be fully determined, but it is suggested that the D-glucose leaving group may be removed by magnesium-dependent acid catalysis. This makes sense because there is magnesium found at the active site of the enzyme. Lactase is released from the alpha-D-lactose molecule by an equatorial nucleophilic attack from water. This nucleophilic attack produces the D-galactose. The second mechanism suggests that glutamate 461 donates a proton to a glycosidic oxygen and a concomitant nucleophilic attack by glutamate 537 occurs. Galactose covalently bonds with glutamate 537. Next, the covalent bond is broken when glutamate 461 accepts a proton, which replaces the galactose with water, thus releasing galactose. Water participation is required for hydrolysis of lactose because it aids in the breaking of the glycosidic bond in the disaccharide to form 2 monosaccharide molecules. It has been observed through experimentation that galactose competitively inhibits lactase activity. Now, isn't that one of the products of lactose hydrolysis? Yes, it is. It has been discovered that the kinetic mechanism of the lactose hydrolysis reaction responds to an inhibition model by the product galactose. Lactase has a similar affinity for galactose and the substrate lactose. The data suggests that the Michaelis-Menten constant and equilibrium constant are nearly the same, which further indicates that the substrate and product have relatively the same affinity to the active site. The graph is depicting the results of the study, showing that there was indeed competitive inhibition between lactose and galactose. The equation shown is the one for the rate-limiting step, which tells the concentration of the enzyme lactose complex that is converted to the concentration of enzyme galactose complexes and the concentration of glucose. The rate-limiting step is the slowest step of a reaction. It is suggested that the breakdown of the enzyme substrate complex is the rate-limiting step for the lactose hydrolysis reaction. The Michaelis constant is representative of the dissociation constant of the enzyme substrate complex in multiple studies. it is possible that the breakdown of the enzyme substrate complex is rate limiting because there is competitive inhibition between the lactase-lactose complex and the lactase-galactose complex. Here's a video showing the location of glutamate 461 and glutamate 537 at the active site of lactase. These two amino acid residues play an essential role in the catalytic strategy of lactase. Lactase employs both acid-base catalysis and covalent catalysis. Acid-base catalysis occurs due to the transfer of a proton. This is seen when glutamate 461 donates a proton to the glycosidic oxygen of galactose. Covalent catalysis is seen during the nucleophilic attack of glutamate 537 at the active site of the enzyme to the galactosyl carbon of the beta-glycosidic bond, which is the electrophilic site on the substrate. This leads to the formation of a covalent bond. Beta-galactosidases are in the GHA superfamily of glycoside hydrolases. All members of this superfamily have a few things in common. 1, they all have an alpha-beta barrel as the catalytic domain, and 2, they have two glutamic acid residues that act as an acid-base catalyst and a nucleophile. So how important is it that the glutamic acids are present at the active site? Will lactase still be effective if glutamic acid residues are replaced by other amino acid residues? An experiment performed by Wan and Huber tested how the enzymatic activity of lactase would be affected if the amino acid residue glutamate 537 located at the active site were replaced by an aspartate, glutamine, or valine. They replaced oligonucleotide primers with the glutamate 537 codon of GAA with GAC, CAA, or GTA. As a side note, experiments have also been performed by substituting glutamate 461 with other amino acids, which yielded similar results as in the study with glutamate 537. These studies indicated that the two specific glutamic acids are important in the function and efficiency of the lactase enzyme. Wan and Huber performed an assay similar to the one I described in my previous video. In their assay, they used the ONPG analog as the substrate instead of lactose, because lactase splits it in the same way it does lactose. The difference with ONPG, though, is when it is hydrolyzed, the ONP product that is formed produces a yellow color. In this experiment, the activity of each form of lactase, that is, normal versus an amino acid residue was changed. Activity was determined by the amount of ONP formed per minute. In this table, the wild type is the native form of the enzyme lactase. E537D is the aspartate substitution, E537Q is the glutamine substitution, and E537B is the valine substitution. As we can see from looking at this table, the mutated forms of the enzyme all caused a decrease in the kcat value. The lower the kcat, the slower the enzyme is at converting substrate to product. The Km values of the enzymes mutated with glutamine or valine were the same as the wild-type enzyme. The Km of the aspartate mutated version was greater than the wild-type enzyme. This indicates that the aspartate mutated enzyme had a lower affinity for the substrate than did any other form of the enzyme. As we can see, when glutamate 537 was replaced, efficiency of the enzyme decreased dramatically. This supports the idea of the mechanism because it indicates that glutamate 537 plays a vital role in helping to initiate the mechanism when lactase does not have the proper nucleophile to perform catalysis. Efficiency of lactase decreases. In summary, lactase catalyzes the hydrolysis of lactose. Glutamate 461 is an acid catalyst at the active site of lactase, and glutamate 537 is a covalent nucleophilic catalyst at the active site of lactase. In an experiment performed, when glutamate 537 was replaced with another amino acid residue, lactase efficiency decreased. I hope this video helped you to learn a little bit more about the enzyme lactase. If you have any questions, please leave a comment. Thank you for watching.